Okay, 2017 paper. This is the multiple choice. Trypan blue is used as a vital stain to identify viable cells when viewed in a hemocytometer. A vital stain, and we're given options of what this is. So this is sometimes counterintuitive in some ways. Okay, what we're looking at here is a stain that interacts normally with either the cell membrane or the cell wall of a microorganism. So if we've got the cell membrane you have to remember is a selectively permeable system. Okay, so this is something that chooses what gets across. And normally what happens with an active membrane is this stain would not get in. It would be blocked and would not be able to get into the actual cell. However, if it is dead and this now no longer has all of the activity that it had before, because you've not got ATP production and various things are not happening, the stain now goes into the cell and builds up, and that's what you see. So what we're looking at here is something which stains dead cells. Question 2. Bovine insulin is a soluble protein with an isoelectric point of pH 5.4. Which of the following graphs represents the level of precipitate formed as the pH of a bovine insulin solution is changed? So if it tells you the isoelectric point is pH 5.4, what that's telling you is that at pH 5.4, that is when it falls out of solution. Okay, so falling out of solution is basically precipitation. So what I'm looking for is at 5.4, I need to have the greatest mass of precipitate, and either side of it, not so much. So that's this one here. 3. The figure shows how a scientist used serial dilution followed by plating to check the number of bacteria in a stock culture. Okay, so here is our stock culture over here. So let me get a pen back up. Okay, here's our stock culture over here. And we are going to do a serial dilution, several serial dilutions, followed by plating out. Okay, so first of all, be very, very careful here. It's asking how many bacteria were there in 1 centimetre cubed of the original bacterial stock. So really what we're saying is we just need to get to here. We don't need to do another move back the way. Because if we go all the way back to the bacterial stock, you're putting in another dilution. It just wants the 1 centimetre cubed. So it's this one here. So I need to get back to this. Okay. So I start off with 119 colonies. Okay. This is 0.1 centimetre cubed, not 1. So that's going to be a 2 in terms of power. Okay, this was times by 10, so that's another one. There's another one. And there's another one. Okay. You'll notice that down here it's in 1.19, so let's just put 1.19 in here and figure out what we did with our kind of bouncing. So that would be 1, 2, 3, 4, 5, 6, 7. That's it. Okay, question 4. Okay, so for this I went and pulled a picture of the sodium potassium pump because you just need to know this really well. Okay, so placing the following events for a sodium potassium pump in the correct sequence. So phosphorylation has to happen after the sodiums have bound. Okay, originally the sodiums have a high affinity, sorry, that's number 3 according to this. Okay, and then we have the sodiums released, and then we have dephosphorylation, and then we have our potassium ions released into the cell. So 3, 1, 4, 2. There we go. I'm afraid you just need to know it like properly well. Okay, another one that I've just gone and got some pictures from to help with explaining it. Okay, we're looking for 2 specific things. The binding of oxygen to hemoglobin is affected by changes in temperature or pH. Which of the following changes would decrease hemoglobin's affinity for oxygen? Okay, so this first one here is showing temperature. So as we increase the temperature, 10, 20, 38, 43 degrees on this one, you will see that the actual affinity is being decreased. So increased temperature causes decreased affinity. Okay, and on this side we've got pH, and you can see here that as you increase the pH going this way, you increase affinity. So decreasing pH is a decreasing affinity. The reason I'm putting it that way around is because it's asking for a decrease here. So I'm looking for an increased temperature and a decreased pH. You have to know these links though, so you have to find a way to remember them. Question 6. Gene represented in the diagram codes for multiple proteins due to alternative RNA splicing. Coding regions are labelled 1 to 10, so these are your exons. Alternative RNA splicing results in an mRNA that contains either exon 1 or 2 or 3, exon 4, 5 or 6, and exon 7 or 8. So basically I've got nothing to do with 1, and I'm not really worried about 9 or 10, because these are the bits that have alternate splicing going on. Okay, so predict the total number of different proteins that could be produced. Right, there's lots of ways you can do this, but this is how I set it out, okay. My options are 2 or 3, or 4, 5 or 6, or 7 or 8, okay. Let's assume that we're starting at 2. So at this point, I have 3 options. I could go to 4, or 5, or 6. But then I have another 2 options on each of these. So I could go 2 to 4, and then I could go 7 or 8. I go 2, 5, and then I could go 7 or 8. I could go 2, 6, and I could go 7 or 8. Okay, so I've got, highlighting here, so I've got 1, 2, going down this way, 3, 4, 5, 6 different pathways that I could take from 2. And I could do exactly the same thing if I started from 3. So basically what I've got is I've got 6 and 6. So my total is 12. In an immunoassay used to detect the presence of an antigen for a disease-causing organism, the pathogen, following samples were tested. We had a sample from an individual thought to be infected, a sample from an organism known to cause similar symptoms but unrelated to the pathogen, a sample from the pathogen, a sample of purified water. Which row in the table identifies the purpose of each sample? Okay, so let's see if there's something that are kind of obvious here. We've got positive control, negative control, and the test assay. Well, the test has to be this sample here. So let's bring them in as a definite, okay? Which also rather nicely gets rid of A and C. Okay, we're now left with, well, it's already given you, in this case, oh no it hasn't, sorry, I thought I'd given you the negative control. So we can get rid of, though, the purified water, because that's not actually involved in any of the ones that we still have active, because we here and here, okay? So we're looking for which of these is a positive control and which is a negative control. So a positive control has to give you a sample giving you something that you expect. So the pathogen is your positive control. So here's our positive control here, and not sure if we're going that way. So that's going to give you the answer of B. And just to be absolutely clear, the negative control, a sample from an organism known to cause similar symptoms but unrelated, because we know that that should give us a negative, hence that's there. Question 8. Which of the following diagrams represents the sequence of phases involved in the cell cycle? You just have to know this, so you just have to figure out which one's going in the right direction. It goes G1 to S to G2 to M, back to G1. And this just cycle goes round and round, so we just need to find one that fits. So where are we going? Let's start with A. So we've got S goes to G1, so that's wrong. We've got B, we've got M goes to G1. Okay, we're doing all right. To G2, nope. Okay, for C we've got S goes to G1, which is wrong, and D goes M to G1 to S to G2. There we go. Which letter in the diagram represents the first stage in cell signalling for a peptide hormone molecule? Okay, so peptide hormone, peptide is the clue here. This will make it hydrophilic. So we have a problem here in that it cannot get through this whole bit here. It can't get through the phospholipid bilayer. So it's definitely not D. Okay, we need it to be cell signaling, so that means that I need something that goes from one side to the other. So it's not B, and it's not passing through because it would not be on this side as well, so it is A. Okay, a calculation question here for 10. Okay, we have red blood cells swell or shrink rapidly in responses to changes in the water concentration around them due to one type of cell channel protein, which is aquaporin 1, AQP1. Each red blood cell can transport a total of 1 × 10 to the 14 water molecules per second, and each AQP1 channel can transport 5 × 10 to the 8 water molecules per second. Okay, how many AQP1 channels are present in the membrane? So it's actually not a bad question. we're just taking, here is our total number of water molecules transported, divide that by how many each AQP1, sorry, lots of letters, could transport, and that will give me my number, which is, or you might have got it in the calculator like that, in which case I expect you to be able to convert it. There we go. Okay, which of the following changes in the rate of the cell cycle could result in a degenerative disease? So degenerative means that we are losing function, so we're losing parts of the cells that are normally there, so it's going to be a decrease for a start. And if it's a disease, this is not controlled. It's got to be uncontrolled. Which of the following diagrams represents the sequences of stages involved in mitosis? Prophase, metaphase, anaphase, telophase. That's your range. So PMAT, B. Okay, question 13. Bacteriophage M13 is a virus that can infect bacteria. When bacteria in the lab are infected with M13, it is essential to use the correct ratio of 1 virus particle to 2 bacterial cells. A 0.25 centimeter cube sample of E. coli culture having a concentration of 8 times 10 to the 8 cells per centimeter cubed placed in a tube. Okay, what volume of a bacteriophage stock having a concentration of 2 times 10 to the 9 bacteriophage per centimetre cubed should be added to the cell sample to give a bacteriophage to bacteria ratio of 1 to 2. Whoa, that reads like quite a lot of stuff, but let's just break it down. Okay, so you've got 0.25 centimetres cubed of something which has 8 times 10 to the 8 cells per centimetre cubed. So if I just do that sum, then I have 2 times 10 to the 8 E. coli. Okay, it needs to have a bacteriophage to bacteria ratio of 1 to 2. So if I take that 2 times 10 to the 8 and divide that by 2, so I've now got 1 times 10 to the 8, that is how many bacteriophage I need to get. So to work out the volume, I'm going to take my 1 times 10 to the 8, divide that by my how many I get per centimetre cubed, so 2 times 10 to the 9, which would give me 0.05 centimetres cubed. That's it. It's not hideous, but it's not the nicest, that's for sure. Okay, a student planned to measure the activity of catalase by using an oxygen probe to measure oxygen production. As part of their pilot study, the student used a standard sample with an oxygen concentration of 22.00% to test the accuracy and precision of the probe. 4 readings of the standard sample were taken. Results obtained are shown in the table. So we've got the oxygen concentration, 4 readings. They are ranging between 20.93 and 20.94. Okay, so let's start with how accurate is being. Okay, so 22.00 was the actual concentration, and this is what we got as a reading. That is not super accurate. That's a fairly major move away from it. It is pretty close to each other. I think these ones are definitely pretty close. So it's not accurate, but it is precise. Okay, so precise but not accurate. There we go. Which of the following formula would allow relative fitness to be calculated? Okay, so for relative fitness, what I'm looking for is the survivors with a particular genotype, the one we're looking at. And then I want to look at all the other ones, all other genotypes. And that will allow us to get a relative decision, a kind of fraction of what's actually happening here. So I'm looking for the survivors of a particular over everyone else. So that is C.